Can the intrathecally be with spinal fusion with rods be administered under radiology?
Hi, for those who don't know me, I'm Jennifer Bain. I'm a child neurologist. And so for individuals who typically have the spinal fusion or difficult scoliosis, we typically do it under fluoroscopy, which means X-ray, to make sure that you're getting it in the exact location. But yeah, that is something that's being done for Spinraza, for spinal muscular atrophy. That's not usually an issue. And then there are other alternatives to getting into the spinal cord as well. So I think there's other ways to get in than just in that one spot.
Thank you. You mentioned, or someone mentioned earlier, forgive me, I don't remember, that the eyes are different. And so if you give intrathecal administration of the ASO, is it something we can expect will improve the optic atrophy or the vision, or we're not sure?
At this point, we're not actually getting into the eyes when we're doing the delivery in this way. And so I don't know that we would do it, but there are gene therapies and other types of therapies for the eye. And I know this is going to sound a little weird, but we do have things where we inject directly into the eye. And so at some point, if we were trying to get the optic nerves, I think we'd have to do an intraocular injection.
Yeah, just you'd mentioned the double-blind study, of course. So after the placebo side would I was a little unclear on whether they would then for sure get the treatment if it was positive. Is that right?
So I think this is hypothetical because I don't have anything I'm putting on the table, but what I'd advocate for is that we would have one period that would be, again, double blind. And then after that, assuming that it's safe, that it would be what we call an open label extension, so that after we finish measuring, everyone could continue on the treatment, whatever that was, and everyone would know about it.
Hi, could you talk about the targeting or tissue specification of the ASOs?
Could you repeat that?
Oh, I was just asking about the targeting or maybe tissue specification of the ASOs.
Tissue specificity of ASOs.
It really ends up being where you deliver it that gives you the tissue specificity. I don't know if that answered your question or not.
I think the slide said after the history trial, after the koala trial, how long does that time frame look like? And then the trial for the ASOs, how long does that trial look like to get? How long you expect it to need to get some answers for the trial?
So this is complete speculation on my part. So right now we have one ASO that works for a variant. We'll figure out who would be able to have that variant and who might be able to use that. At that point, it's a discussion with the FDA in terms of what they'll allow us to do as the next phase is going forward. I don't know exactly what they're going to say. I think that's the next discussion. I think it's important for everyone to have, anyone who would do this needs to have baseline data so we know something to compare to. So that was the point in koala, is whoever needs to do that would have those baselines so we know where we're starting. Anyone could do that. The more data points we have, the more sure we are of where we're starting. But in theory, you know, it could be a single data point that tells us running into it.
I got asked to moderate this, but this is a really good question. Jen, can you say it?
I just want to make sure, you know, I think ideally you would continue the natural history study regardless. It's not consecutive, just to clarify that. You would keep collecting data about natural history with interventions along the way. And that, just to make sure everybody's clear about that. It's not like you're waiting for koala to end and then you would do the clinical trial. Yeah. Okay.
So is the vision that this will be multiple
single patient protocols, or would it be a clinical trial for participants who have a particular genotype that will be amenable to the ASO?
So this is pure speculation on my part. The FDA is not going to let us do N of 1 trials forever, right? At some point, they're going to say, you need to do a real clinical trial. You need to be blinded. You need to be rigorous. I don't know how many there, at what point they say that we're going to need to do that. I just don't know. And we haven't gone to them yet with that question. You could imagine that for each ASO, it goes through the same process. You could imagine there could be combination ASOs. I mean, you can think of all sorts of things in terms of going forward. I think from my point of view, in a good way, there is potentially a path going forward. And I think we need to collectively figure out how quickly we can move forward. That's not in a reckless way, that's in a safe way. And pretty soon we need to start engaging the agency in terms of what they're going to allow us to do.
If I could just jump in as a moderator and ask 2 questions that I've heard from at lunch. People have mentioned some terms and some questions. One is maybe Pat and Wendy, and you guys can touch on the handle effect or the handle approach rather, not effect, and how that might be a hopeful treatment. And also somebody asked me what an IND is, and I think you all are far more capable of answering that than I am.
Oh, I got one. So maybe I'll start with the IND question, because that's probably more my specialty. So an IND is investigational new drug application to the FDA. So that's the terminology we say for the first time we want to test a drug in a person. We apply to the FDA with a lot of data, right? This is it's a bit of an exercise of documentation, but really what it comes down to is safety is the primary consideration when opening an IND. Is our compound, in this case an ASO, safe to be administered at doses in people that we understand from animal studies? And typically we test our drugs in 2 different animal species to make sure we understand the safety. And for an ASO, it would most likely be a rat and a monkey, or a cynomolgus monkey. And we'd understand really how high we can push the dose before we see toxicity, so that we can use that scaling to people and understand the safety margins. I'm guessing, as we move forward, the agency, FDA, is going to ask us to do those studies for these ASOs when we start applying this to more and more people. And so on top of that, we typically look at off-target scenarios. Like I think Wendy was saying, a minute ago, toxicity with these ASOs could be target directed or non-target directed. So off-site, and there's lots of different techniques we can look at that. So that's typically called safety pharmacology. PK is another aspect of that IND. So how long does a drug, once it's delivered, stay in the body? And how long does it take to be eliminated? And then obviously, if we can inform the agency of how long we think the drug will last. So that's the pharmacodynamic effect. So that's how long it's on target doing its job by knocking down the mutant form of the KIF1A in this case. And so all the information would be have to be generated, I think, experimentally, and that can take a little bit of time, but we kind of know that process. It's relatively formulaic. So that's an IND. In any, typically the agency asks an IND for one compound or one entity. I think there's a lot of conversations now in the world that when you're talking about ASOs, if they're similar, that you can start considering them as similar drug entities, but that hasn't happened yet, where we can just say, well, ASO1 and ASO2 can be filed under the same IND. I don't think we're there yet, and those conversations are happening. So maybe Wendy, you want to talk about the handle again?
Yeah. So the idea, again, if you remember those dots on the screen, is that we would like to take a shortcut in the sense of, rather than having to make an ASO for every single different dot, can we lump them together? And so the idea is that there are common genetic variants among us that happen to be on the same strand of RNA as the problematic KIF1A. And so if there are small, and I'm just going to say 10 to 20, but a small number of those handles that'll cover the vast majority, if not everyone, then we don't have to have 100 different ones for all the different mutations. We may simplify that and make it 10 or 20% of, what we otherwise would have had to do. So within that, we've got to do the inventory in terms of this. Anyway, I won't go through all the algorithms of how I've thought about doing this, but I do think that flexibility will be an important thing for us to do.
Can I ask two more questions that came from a lunch conversation? These two questions, Wendy kind of touched on one of them. And then the second one is a really, really great question is when we are talking about what an ASO targets and if we are seeing something that wasn't an endpoint, right? And would a clinical global impression be an endpoint?
Yep. So what Luke is pointing out to is, I think of it as what Dr. Bain and I would probably be our overall clinical impression. And so it's just this, as a parent or as a clinician, you just know it when you know it, right? In terms of things getting better. And it's more rigorous than that. I don't mean to be pejorative, but it's overall just, does it look like things are getting better? Does it look, and you use that as an overall impression. I have to say it's squishy. So we use that all the time, but it's squishy and it's not the most rigorous of the outcome measures that we use. So every clinical trial has some version of this in it. But I do think they're in a world that's increasingly quantitative and data driven. I think we can do better. And I think the more that we can, the biggest thing for me is sensitivity to change. Can we, in a rigorous way, see small changes so that we don't have to wait months to years for large changes that are in the usual tools that we use?
The second question.
Well, can I just say one more thing along that? I think it comes back to this term biomarker you hear all the time, right? Is biomarkers kind of exist in two ways in my mind. There's clinical biomarkers where these clinical changes you can observe in a person live, right? And then there's the molecular biomarkers. Those are the things that we can measure in body fluids that tell us that the drug is doing something and things are changing within that person in a positive way and indicating that that drug is doing what it's supposed to do. So coming back to why you're all here is eventually your samples here will set the course for biomarker understanding in our clinical trials, looking for those changes. So I think it's a really important aspect.
I'll just add that, not only do you need to look for change, but in clinical trials, they're not 5-year studies, right? In general, they're going to want to see smaller changes more quickly. And then importantly, while you're all here as well, it's important to hear voices from families to say what's clinically important to you. This isn't, and I don't want to bash people who study hypertension or high blood pressure, but they want to measure a medication or an intervention. Let's bring everybody's blood pressure from 200 to 120, right? This isn't that simple, right? So CAND is very diverse. It's very heterogeneous. Everybody looks a little bit different, but there's overlapping features. So, what are the pieces that we can measure? Are we just measuring vision? Are we just measuring seizures? Are we just measuring motor skills? And so those are all big pieces to this. What are the things that are most clinically impactful? And the FDA will look for that as well in terms of, making sure that you have your patient voice in there for FDA approval.
Yeah, that's another thing that we will all talk about in a different time is how we can impact FDA. I think that's a big community discussion. And we can have guidance certainly from these incredible professionals sitting next to me. But the second question that somebody asked at lunch was, Wendy spoke to this, but I wonder if there's more to it, and I'm not sure. When you spoke about SMA, the question was, how do you take your learnings and skill sets from other diseases to apply them to different diseases, including KIF1A? And I know that.
Yeah. So there are a lot of learnings from SMA, a lot of differences as well. But the things that are similar, for those of you who don't know, SMA is also a degenerative condition. And so FDA, I think, thinks about those conditions a little bit differently. The ASO was also helpful in a very different way. So the molecular mechanism very different between the two diseases, but the ASO very helpful as the first of the treatments for that. And I won't go into all the details, but we ended up playing a very critical role in terms of newborn screening for SMA. And at some point, some other day, I'll talk to you about newborn screening for KIF1A as well. But I do think there are similarities there. SMA also relatively quickly went from an ASO requiring repeated dosing to a gene therapy. And the gene therapy for SMA is given as an IV infusion and is theoretically a one and done treatment. I won't talk about what the long-term effects of that are because I don't think we know yet. But the hope is that the ASO acted in my mind, in part as a bridge and a backup for another strategy in addition to the gene therapy. And I do think it's good to have more than one way of treatment when it's possible. And I do hope that we eventually have something that is a one and done type of treatment. I don't know that it's going to be an IV infusion. I think we're going to have to go directly to the brain. But I do hope that we have something that's more permanent than an ASO. I do think there are a lot of things to learn from other rare disease organizations in terms of just how to get things done, how to interact with government, how to interact with agency, how to interact with payers and insurance companies. I think there are lots of lessons learned in multiple dimensions. But I think the one that's going to be most important is actually the delivery system. So I talked about this before, but getting into the brain, getting across all the cells in the brain, getting evenly, not getting too much, not getting too little. There are some tricky things that go in with this. And the good thing is that we're not the only brain condition. There are hundreds of other brain conditions out there, many of which affect many more people. And so that's good. And so we're going to learn, I said it before, but we're going to learn from Alzheimer's, Parkinson's, Huntington's, any number of other diseases that are also working on this as well.
I know you dosed hundreds of people with ASO. Are there learnings that you take from?
I think, you know, I got into this with SMA about almost 9 years ago. And so we feel very comfortable from a research and from a research, is it on? I'm not speaking into the microphone. That's helpful. So I think the nice thing is that we feel very comfortable and confident medically that many different professionals, not just one person, can do this actual administration. But similar to the question, I think earlier and what Wendy just mentioned, that, you know, we're going to learn more about which cells matter and maybe what specific nuances to the delivery are going to be important, but the actual procedure itself of a lumbar puncture with intrathecal medication is something that's been done for decades, even with cancers, and way before ASOs for rare disease. So I think that method of delivery, we've effectively said, yes, we feel very comfortable doing this. We know how it works. ASOs are certainly an expanding platform, but it really is number one, safety, and then second to that is efficacy after that. So I think you're in a good position that there are many other diseases out there, rare diseases, that are using a similar platform. And so learning both what went well, but also a lot of what didn't go well is going to be really helpful along the way. It's something Patrick always says, he didn't want to say it, but he always says more shots on goal.
Oh, yeah. Yeah, exactly. With ASOs, we have a potential for many shots on goals, but we want to keep it simple though, too, right? Fewer would be better in this case.
And I think the reality is, you know, at least when I started practicing, there were no treatments for spinal muscular atrophy. And now we have 3 options. And there's still different clinical trials and different treatments, and it continues to catapult forward. It didn't just eliminate the field of SMA and SMA treatment. And, you know, I think that's exciting because you haven't even gone into the next phase of treatment and standard of care.
Yeah. I think there's over 30 ASO trials running right now in the US alone. And I think we'll learn a lot from them. Angelman is one close to my heart because of Ovid, and we've been involved in Angelman in the past. You know, Roche actually shut down their trial, which is unfortunate, but Ultragenyx is continuing. And again, they took a step forward, had some problems, took a step back, reconfigured, and now I think they're really on a good pace to show efficacy. So, you know, every trial matters for these technologies.
Thank you. So speaking of multiple shots on goal and also of trying to have parallel processes that are contributing to each other, you know, you've spoken about how gene editing is going to be a more permanent potential solution than ASOs, and there are some advantages and disadvantages to that. But I wonder, with ASO targeting, is that a lesson that we can then apply to prospective gene therapies when we're talking about how do we actually target the mutations that are present in our community, and how do we do that in an effective manner? And I was just wondering if you could speak to how targeting translates between these kinds of modalities, or whether it does.
I'll make a comment that So SMA uses a very different technology and a very different mechanism. But if we look at the muscular dystrophies with gene editing, so muscular dystrophy or Duchenne muscular dystrophy, also a degenerative muscle disorder. And so interestingly, there's a lot, dystrophin is like this really massive, actually our largest protein, and there's lots of different causes. So lots of different gene changes similar to CAN, that you have lots of different changes as opposed to other diseases where it's really just one main switch or change. So with the muscular dystrophy, similarly, a couple of years ago, there was nothing. It was just symptomatic treatment. And now they're getting to the point where they're having specific kind of chunks, I guess, a similar handle effect to saying, can we kind of maximize the percentage of people that we can support. So I think for gene therapy, that's moving towards a, clustering event. And it's not just one specific treatment. It started with just one gene editing, and now it's catapulting forward to kind of getting larger and larger chunks of the population. So getting to know people who are being affected and how it's affecting you in a setting like this is just really critical to saying, how can we look at the larger group? How can we get a bigger bang for our buck in terms of treatments? If that answered your question.
Yeah, thank you.
Yeah, I think what's interesting is when we started this project, I think with Wendy's group and Charles and Michael probably can remember this a little bit, is we kind of were hoping for the holy grail in a long-term solution. So we started our projects thinking that AAV delivery to the CNS, delivering a sequence that would permanently, change or have permanent effects. We took a step back because that was really challenging. But any ASO, I think here, any sequence that we develop as a community that is a temporary treatment, so every 3 months you got to deliver it, could be, in theory, and I think we're very optimistic, converted into a long-term permanent solution. So when the technology becomes a little bit better, Zolgensma is the example that we all hear about. AAVs are very tough. They're very expensive to develop. There are some more risks associated with them, but I think down the road, we could have a permanent solution, and these ASOs will inform that, right? So any ASO can be moved into a vectorized version and be more of a permanent solution. So this is enabling information. It'll lead to the next generation medicines.
Yeah, so because it's so complicated with CAN, like people can have the same defect in the gene, but present completely differently. Is the ASO therapy entirely based on the fact that it can grip a specific handle sort of in that genetic sense, or is the phenotype kind of considered in that eligibility or possibility of it being effective?
So I have to admit, I don't think I completely heard the question, but from what I heard, there is quite a bit of variability clinically in severity.
Yeah.
As we think about the treatment really is focused on the genetic variant itself. It has to be focused on that. The outcome measures that we'll use to measure whether or not we're having an effect will take into account severity and the age of the person to try and just see if we're moving the needle. Did that answer your question?
Yeah, I guess so. It's more like, so it has to be based, obviously, I'm just trying to understand that chemistry side of it. So it has to be based on the fact that it can grip onto a specific handle versus the fact that you could get two different children that have very similar presentation, but present very differently in their mutation.
Right. So the thing that makes this a little bit sort of a mind bend to do is that there is, by the mutation in KIF1A, the thing that's actually causing the disease, there is a correlation between that disease associated variant and the severity of the disease. The handles are not at all correlated in any way with the disease severity. And so we could have one handle, the same handle that'll work for someone with mild disease, and also that same handle will work for someone with severe disease, right? It can go across the full spectrum of disease severity. But what we would use to measure the impact would be different for someone with severe disease, than it might be for someone with mild disease. So we're going to have to think about that as we design and look at the impact. And that makes the clinical trial design much harder than it would normally be.
Yeah, that's
You're right about that.
Yeah.
I'll just add that logistically speaking, if a company or a sponsor is interested in a treatment, we all want it to work, but they really want it to work. Okay. And so what we often will see, what we have seen is, even if we see that something is working, we need to make sure it works enough. And so very often, and this might come into play in the next, steps in terms of who would be part of what they call eligibility criteria, right? Like who are the people we're looking for to see those small changes that are meaningful? Because unfortunately, if you have somebody who's very mild and maybe only makes a small change, that might not be as impactful or as powerful to somebody who's sponsoring a project to say, as opposed to somebody who's more severe and you're seeing a larger change. So it becomes a little complicated in terms of how do we measure that change? So there's the trial part and then there's the treatment part later, but showing that, yes, this is a treatment or an intervention that's going to work is a little bit different than like later down the line saying, yes, this treatment could be applied for everybody. At the beginning, it really is going to be very careful. We need to actually say that it's actually making a difference and making a change.
So if I understood, there are definitely people that have the same mutation. I don't know which people have the same mutation and the same handles at this point. That's the information we're putting together, but definitely people who have the same primary mutation that associates with CAN and the same substitution. Those people tend to look very similar clinically. They're due. And for those of you, there are a few of you that where there might be even 20 people with the same KIF1A mutation that's disease associated. There's a little bit of variability, but you tend to look similar to us.
Thanks. I did one follow up on that. So are you saying that everyone with the same variant has the same handle, or we're not sure of that?
So it's indubitably the case that people with the same, and I'm going to use the term mutation, KIF1A disease associated variant, do not have the same handles. Okay. It's going to be just a random thing, which handles which mutations.
Okay. And is there a possibility, are you thinking about doing another N of 1 that would be handle based as opposed to variant based?
It is possible that if another handle can be developed, I don't see a reason not to try to develop additional handles, and that could work for whoever it works for.
Okay, thanks.
Thank you. I have two questions. First one will be on ISO in general. I understood that we can knock down a mutant KIF1A to prevent the protein from being created. But with an ISO, can you fix the behavior? So having it created, but make it work? And my second question is also on ISO. Today, it seems expensive to create a new treatment. Could we expect the cost of such a development to decrease in the years to come?
I think if I understood the question, the ASO is not going to fix, I think was the term you used, either the gene or be able to convert a dysfunctional gene into a functional gene. The strategy that we're using is it kind of silences or takes out the garbage is the way I think about it. There are ways of making ASOs that will help increase the amount of gene that's produced, but that's not the strategy that we're using in this case. And then can this get faster or easier? Was that the second part of the question over time? Cheaper. So I would say the manufacturing cost of the ASO is not the main problem. Actually, manufacturing, I'm not saying it's cheap, but it's not the biggest thing. I think it's all of the development process that goes and then the toxicity studies that have to be done to make sure that it's safe. That's where the expensive part comes in. And I don't know, Patrick, you may know better. I don't know easy ways to scale that.
No, I mean, they're typically just standard studies that the FDA asks us to run in a set number of animals and evaluate. So, and for this, we have to look at longer term toxicities, right? So since we're going to be dosing people for years, we have to do long studies in animals, and those are typically expensive studies. So there is some initiatives too to kind of push away from using animal studies to do predictive toxicity. I wouldn't recommend doing that quite yet. I don't think we're there. But along the lines of editing though, so there is some new technologies, and it's not anything we're working on, but there are technologies coming out that are based on RNAs like ASOs that can do editing. It's not like CRISPR in the sense that CRISPR uses a guide RNA and some enzymes to make these changes, but but there's actually some technologies that are really exciting and hopefully will come up in the next couple of years that will allow ASOs to do editing. But that's down the road. So exciting technology, but not quite ready for prime time.
Thank you. This is a question concerning strategy. So what you're doing right now is you construct an ASO to silence the mutation, right? Would it be feasible, or what is the rationale against putting in a healthy protein to sequester away the mutant protein? Is that a feasible way?
I think there's some evidence that overexpression or upregulating KIF beyond its normal expression can lead to toxicities in it itself. And so we're just trying to get rid of the toxic gain of function is really the key component here. Is that right, Wendy?
I think it would be remiss not to mention N-Lorem in this conversation. And I would, we talk about manufacturing and about the people who are developing the actual product or the actual treatment. And N-Lorem is an incredible foundation that set out to do this for people and develop the treatment for free. But the cost, like Wendy was saying, was when the baton gets kind of handed off. And that's a big cost. But N-Lorem is something where you should apply for it. They have an entire brain trust of people who are looking to see, is this mutation ASO amenable? And they work with CROs, they work with other folks, and they're really good shepherds and really wonderful people. So I would say we've shared it with the group, but if you haven't gone on to N-Lorem's website, they also have a lot of really great resources about, explaining in a very colloquial way the science behind things that I certainly it's above my head. But anyway, so please do check out that website too.